Certainly. So I originally trained in Australia at Sydney University, where I did a Bachelor of Science Medicine degree and a medical degree. And not long after finishing my internship at Royal Prince Albert Hospital in Sydney, I went to the United States to work at a place called Cold Spring Harbor Laboratory, which was directed by Jim Watson of Double Helix fame, and worked on transgenic mice and gene regulation in cancer. And from that experience, I decided that I wanted to apply that genetic manipulation technology to study the brain and fundamental questions of behavior, and in particular, how learning and memory worked. And I went to Columbia University in New York City after finishing a postdoc at Cold Spring Harbor. And there I worked with a man called Eric Kandel, who was studying a marine invertebrate, studying learning and memory. And I worked in his big group there, doing gene manipulation in mice to study learning and memory. And that was in the early 1990s. And that kind of work became extremely popular after that, because it was a very powerful way to discover how genes are important in learning and other forms of behavior, because we could directly manipulate the genes. As part of that work, I focused my attention on synapses, which are the junctions between nerve cells in the brain. And in the human brain, there's about 1 million billion of those. And within those synapses, at the time I started this work about 30 years ago now, there were very, very few proteins that were known to be parts of synapses. I became interested in understanding those proteins and the genes encoding them, and focused particularly on the proteins that are on what's known as the postsynaptic side of synapses. That's the side on the receiving side of the synapse. And that's where the information first comes into nerve cells. When we started to use a method called proteomics, that there were very many of these proteins in the synapse, and the synapse was in fact much more complex than what we and indeed everybody else had previously thought. And so over a number of years, we worked on those proteins and how they were complex, that is to say many different types, and discovered many of them were very, very important for lots of different human diseases. And in the last few years, another direction that's come from that work has been to realize that those proteins are not all found in the same synapses. They're sort of scattered around in different combinations in the different synapses of the brain, generating diversity of synapse types. And traditionally, we've thought of synapses as being of really two major varieties, what's called excitatory or inhibitory. But it turns out that within each of those categories, there is vastly more types of synapses than and we don't even know how many types there are. Now, to look at that problem, you need to have ways of studying the proteins inside individual synapses in the brain. Now, an individual synapse is pretty small. It's smaller than a sort of a cubic micron. And one way to do that is to use microscopy methods, whereby you label the individual proteins and you look at the synapse and ask how much of the protein is found in that particular synapse. And over the last few years, we had started to develop ways to look at that, and had published a paper in the journal Neuron in 2018, where we showed for the first time that you could do this in a very large scale way. And we examined synapse proteins across the whole areas of the mouse brain, and therefore could in fact do a survey for the first time in any species of the molecular composition of individual synapses across the brain. And what we found was that there were very many types of synapses, more than we had thought. But not only that, there was not just diversity, there was something that was really surprising, is that the different types of synapses were spatially distributed in a patterned way across the brain. And we refer to that as the synaptome architecture. Synaptome means the diversity of types of synapse in the brain, and the architecture refers to their spatial organization. And it was this remarkable, I think, finding that there were the synaptome architecture of the brain that led to the application for this new ERC grant, which I began to write for in 2019. Now, in that proposal, which has the name Synaptome, what we wanted to do was to understand how this remarkable patterning in the brain occurs and what are the mechanisms beneath that. And we realized that it was extremely important to study that at what you would call the single cell level. So in any individual nerve cell, there could be as many as, say, 10,000 different synapses, sometimes more in some cells. And we wanted to look at the synapse proteins inside just individual nerve cells. And the brain is also made of many different types of nerve cells. And so it would be sensible to think that perhaps different nerve cells have different compositions of synapses across their branched trees, known as the dendritic tree. And that's what we wanted to study. And so we've come up with methods to do that. And we wrote a very focused proposal to the ERC to study what we call the single neuron synaptome architecture. And we focused the project directly on doing some very basic science in this new area. And to our knowledge, there's nobody else doing this kind of work in the world at the present. And we were in an extremely good position to be able to do that. And I think that's one of the reasons the grant was successful. Well, I already had an ERC Advanced Grant already, and that grant was due to expire in the middle of 2020. And so the normal thing that one would do in such a situation like that is to attempt to be successful a second time. And the time that one can apply for those grants had a particular time window that was attached to when your existing grant actually expires. Now, the problem for me was that being in the United Kingdom and this grant coming from the European Union, the United Kingdom was in the throes of the Brexit negotiations. And I think everybody will realize that there were a great deal of difficulties in those negotiations, and that the deadline that the UK had imposed kept being pushed back. Now, oddly enough, that turned out to be a great advantage to me, because had we left the European Union, it was uncertain if it was going to be possible to make an application to the ERC again. And so at some point during the British negotiations with the European Union, one of the delays occurred to the point at which I was then able to make my application within the time window left in my existing grant. It was a 12-month time period. And so I was very grateful for one of those extensions. I suddenly realized that if I get my act together and write a grant application, I might be able to win another ERC grant. And so I wrote that in the middle of last year, in 2019, and then made that application. Now, as far as anything that was particularly challenging, I would say that the most challenging thing was actually getting the opportunity because of these very unusual political circumstances. But that's not the kind of thing that applies to other people here. And so I suspect they won't be very interested in that. I think that the challenge in putting together an ERC grant is that it's very clear that they want something that is adventurous and exciting and different. And in my experience, the first time I wrote for an ERC grant, I've written for 3 of these. The first time I wrote for an ERC grant, it was the sort of grant that I might have written here in the UK to one of the funding agencies like the Wellcome Trust or the MRC. And it was what I thought to be a particularly good grant. It was strong and conservative. And I emphasize conservative in the sense that it was based upon lots of things that we had been doing recently and looked very solid and doable. And we showed lots of preliminary results and that kind of thing, and published papers. But when I first sent that into the ERC, they said, that's all very nice. And that looks like very much the sort of thing that you've been known to do over the last, 5 or 10 years of your career. We would like to see you do something a bit more exciting and adventurous than that, and please resubmit a new proposal. And so I resubmitted a new proposal, which had entirely new stuff, which at that time was this work on these synapses and the molecules and the imaging of the brain. None of it was published. And I was successful in that grant application because they liked the excitement and the novelty. Now, and the same thing is true when I wrote this second proposal. I made sure it was something that was very exciting and doable, but it wasn't something that we had been doing. Now, that is a very interesting and challenging position, because that would not be the way to get a grant successfully funded in the United Kingdom. And one of the things that I've been very, very pleased about with my experience with the ERC, and indeed other European grants, is that they have taken a much more adventurous view, in my mind, than what the grant funding agencies in the United Kingdom would do. So I always make sure that when I write a grant in the United Kingdom, it's a more conservative grant, and a much more adventurous grant would go to the European Union. The benefits of the ERC advanced grants really are that they really allow you to do those adventurous areas of science. They're not concerned as to whether or not it's going to be useful. It's not driven by sort of political pragmatism in whatever form, in saying that it's going to be useful for industry, or it's got to be useful for medicine. It really sticks to the fundamental basic principle that good, high quality, basic adventurous science is going to pay dividends. And there's a very long history that makes that very, very clear that that is the case. And so it's a very safe bet. And that's a very good thing, because many people who are not scientists think that the way to fund science is to tell them that they've got to solve a particular problem, like cure cancer or land on the moon. But as has been pointed out by many people before, that's really an engineering problem. That's not a scientific problem. And so what I like about these and the benefits of these is that they give you the space to try to do something, you know, really original, that's not focused on any particular outcome. Well, you should go for it and put something together that's good, adventurous, and high quality. And that's going to go into some new direction, and be bold about the whole thing, and make sure that it's a very strong application where, as much as possible, you have a strong track record. You know, make sure that that is clear, although the reviewers will be they will certainly work out if you have a strong track record or not. But the point is to make a good, solid, adventurous grant, and don't make it boring.